when it comes to white mold, or the Latin name sclerotinia, sclerotiorum, it is canola's #1 yield killer. That means that growers from all across Canada have to deal with sclerotinia from Manitoba through to Saskatchewan as well as Alberta, and represents a huge economic loss to growers right across this country. When you think about canola, you think about these beautiful fields of gold, not only surrounding the University of Manitoba in Winnipeg, but all across Canada. Canola represents an almost $30 billion industry to Canada. It represents almost 250,000 jobs. We probably consume canola oil in one way or another, whether it was extracted from the seed or other means, usually every day in the food that we eat. But it's not only the really heart-healthy canola oil that is so great for us, but we're also finding new and innovative ways to use the plant material when it comes to biodiesel, or when it comes to producing different types of cosmetics, or when it comes to producing the next generation of textiles. When it comes to finding new and innovative ways to protect Canada's #1 crop, we turn to genetics because it represents a safe as well as affordable way in protecting the crop. If we cannot find new and innovative ways to protect canola, we can in fact lose upwards of 30% of the overall yield in any given year. That's because we are seeing more pressure from a changing climate in terms of how those different types of adverse environmental conditions might be affecting the crop. We're taking a really novel approach to protect canola against white mold, or sclerotinia. Traditionally, growers have access to broad spectrum fungicides, and these interfere with one of the primary cellular metabolic processes of the fungus. One of the concerns when it comes to these fungicides is that they also might have negative impacts on other beneficial organisms that are found within the ecological environment. But what we're seeing now is increasing need to find these new technologies, because we are seeing more resistance that's developing in sclerotinia, not only here in Western Canada, but all around the world. The idea that we have in collaboration with Dr. Steve Wiard in the Department of Biological Sciences here at the Faculty of Science at the University of Manitoba was to develop a species-specific molecular fungicide. So that means that we're targeting sclerotinia alone. We have no off-target effects, at least to the best of our knowledge, and we're not seeing any negative impact when it comes to the environment. So this type of technology really represents the next generation of control methods that can be used in order to manage canola across Canada. Some concerns, especially when we think about how some of the first generation of transgenic plants, or the use of genetic material to be able to make a better crop, has really been communicated to the general public. We really dig deep into the literature to make sure that the molecules that we are working with, and we're working with these double-stranded RNA molecules, are species specific. We've also done a number of tests to make sure that there are no off-target effects. And we also do a number of experiments to show that these molecules can degrade within the environment. We've been working on the use of RNA biology to better understand ways of managing crop pests as well as pathogens for over a decade, in collaboration with Steve Wiard. And now we have gone through a number of really rigorous lab as well as greenhouse experiments. We've also started to do a number of field trials, and these field trials, in collaboration with the Pest Management Regulatory Agency of Canada, will provide us with the insight that we need in order to show that this type of new technology is going to be effective out in the field. It's really only a matter of time before we see the similar registration of these next-gen technologies right here in Canada.